Good evening, everyone. Welcome to our course. I am the editor of the Bhekisisa Center for Health Journalism, and we are very excited about hosting you tonight and in the other sessions that we're going to present. And we thank you all for registering and being excited about our course too. And we really look forward to hosting you. We've had close to 100 journalists registering for this course. And this, of course, follows our course on Epidemiology 101 for Africa journalists that Bhekisisa and some other partners hosted in July last year. And if you're interested in looking at that course, there's a link in the chat box to the course. For this vaccine course, Bhekisisa has partnered with the Center for Evidence-Based Healthcare at Stellenbosch University, and they have coordinated the health specialists and the vaccine experts who will be speaking and lead our sessions. And I'd really like to thank the director of the Center for Evidence-Based Healthcare, Taryn Young, for all the work she's done on this course. And without her, we would not have had speakers or a curriculum for this course. So Taryn, thank you very much for your dedication to doing all of this. Our course this time around will focus on how vaccines work and how we know if they're effective and safe, and also the challenges with distribution and how variants will affect the efficacy of vaccines, and also how vaccines are procured and how COVAX will fit into all of that. We will allocate a large part of each session to answering questions from all of you. And for this, you can enter all your questions in the chat room or box. And Anel Skoenius from the Center for Evidence-Based Healthcare will collect them and post them to our speaker tonight, Hassan Mahomed. And you don't have to wait until the end of the session to enter your questions. Anel will collect them throughout the session. So as soon as they pop into your head, you can just enter them into the chat room. I've got two things to ask you before I'm going to hand over to Taryn Young from the Center for Evidence-Based Healthcare. The one is that we've sent you all a course survey to fill out when you registered, and we've received quite a few responses, but from some of you, we still need a response. The link is in the chat box, and we'd really appreciate it if you could fill it out. It's only 10 multiple choice questions, and it takes 3 to 4 minutes to fill it out. So if you're able to open the link and fill it out, we'd really appreciate that. Then I'd like to ask all of you to please make sure that you mute yourself fully during the session. Otherwise, there would be disturbant noises that interfere with the speaker tonight. And we'd also appreciate it if you turn off your video so that we can focus on the speaker. The speaker will have his video on. And then I'd like to hand over to the director of the Center for Evidence-Based Healthcare, Taryn Young. She will introduce our speaker tonight and also the subject he will focus on. And she will also tell you a little bit more about her center. Taryn, over to you.
Thanks, Mia. And it's really fantastic to be here tonight. It's really exciting that we have this course and can be collaborating with Mia and the team. I'm based at Stellenbosch University, where I head the Center for Evidence-Based Healthcare, as well as our Department of Global Health. And we've been very active in responding to the COVID pandemic in terms of conducting research and promoting the use of research in decision-making. It's fantastic to have my good colleague, Professor Hassan Mahomed, with us tonight, and he will cover the session on vaccine development and also more details around how vaccines work, and specifically to COVID, which vaccines are available, and also the whole process of getting vaccines registered for use in country. So Hassan is a public health specialist that's working with Stellenbosch University within the Division of Health Systems and Public Health, as well as with the Western Cape Department of Health. Hassan has been very active with the COVID response, and he's also the chairperson of the expert committee on COVID vaccines. So to not keep you waiting further, I'm handing over to Hassan for this evening's presentation. Hassan, over to you.
Thanks so much, Taryn, and it's a great pleasure to be able to present this session tonight. I think let me just start off by saying I've kept it fairly basic at the moment. Always never easy to know exactly how to pitch a presentation like this. So I've kept the talk as jargon free as possible and as basic as possible, but please feel free to ask any questions that you have. Just share my screen and let me get into, okay, perhaps Taryn or Mia, just confirm that you can see my screen.
Yes, Hassan, we can see it.
Okay, thanks so much. So let me get started. So just for yourselves and for myself, what we want to achieve today is to enable you to understand how a vaccine works, how vaccines are developed, how vaccines are tested, and how they are approved. So I wanted to start off, and I think this is familiar territory for all of you on the call, is about sources of information. And as you know, many people talk about the misinformation pandemic as well. So these are sources that I use. So let me start off by saying that. So in South Africa, there's the National Institute of Communicable Diseases, and they have an excellent website providing a whole range of information on COVID and on vaccines. And then, of course, internationally, the World Health Organization, always takes a global view. And there's also, of course, an Africa office if you want Africa-specific information, but there's their website that provides information. And then, of course, I also go to the Centers for Disease Control, CDC, in the USA. They produce information which they make freely available to the world, and I found a lot of the information very useful, a lot of it very, public orientated. A lot of the information I'm going to use in this presentation comes from the World Health Organization website. So it's something that you can go back to and check, but of course this presentation will be made available to you as well. So starting off with just some of the basics, like how do vaccines actually work? So vaccines are simple, safe, and effective way of protecting people against harmful diseases before they come into contact with them. So important point to remember there, it's to prevent you from developing a particular infectious disease. And again, an important point to mention is that it uses the body's natural defenses to develop resistance to specific infections and to make your immune system stronger. So it's really about what our own bodies are capable of doing rather than, the vaccine itself, but it's the ability of our bodies to create defense mechanisms that's utilized through these vaccine approaches. So vaccines train your immune system to create something called antibodies, just as it does when it's exposed to a disease. However, because vaccines contain only killed or weakened forms of germs, they do not cause disease or put you at risk of complications. So just to emphasize this, when a virus or bacterium or any organism that causes disease enters our bodies recognize this as foreign or not belonging to the body, and our bodies then develop an immune response. One part of that immune response is to create these proteins called antibodies. And the antibodies are very specific. So you have an antibody for a specific organism, a specific virus or specific bacterium. So they are developed in quite an amazing way by our bodies. And there are specific parts of the germs of the viruses or bacteria that we develop antibodies to. And these are called antigens, and they're usually often proteins on the surfaces of viruses. And they are commonly known as antigens. And then our body's defense against these antigens or organisms is in the form of these antibodies. So the antibodies fit against the antigen like a lock and key. So there's a very specific fit. And I'll come back to this, a little bit later. Most vaccines are given by injection, but there are some given orally or sprayed into the nose. So there's a range of ways of getting a vaccine, but most commonly it's given by an injection, usually into the shoulder. When you get a vaccine, what does your immune system do? It recognizes the invading germs, such as the virus or bacteria, produces antibodies. The antibodies are the proteins which are produced naturally by the immune system to fight the disease. And this last part is extremely important. It remembers the disease and how to fight it. So there's this interesting thing called immune memory. And that's really the fundamental basis of vaccines. Vaccines would be useless if our bodies were not able to create this thing called immune memory. So the body sees this piece of germ that's harmless in the form of a vaccine. It produces antibodies against that vaccine, and then it stores this memory so that when the actual germ comes along, the body is ready to respond. The importance of this is that when we first exposed to a disease, it takes 10 to 14 days before our bodies develop antibodies, so it takes time. But once you've got immune memory, when you're exposed to a virus or bacterium that causes disease, your body responds quite quickly within a day or two. So exactly as is stated here, if you're exposed to the germ, your immune system can respond quickly and destroy it before you become unwell. So this vaccine is a very safe and clever way to produce an immune response without causing illness. So the vaccines themselves do not make you sick. They cannot cause the disease itself because it's usually a piece of germ or it's a protein. And I'll explain later the different kinds of vaccines that you can get. Our immune systems are designed to remember. Once exposed to one or more doses of a vaccine, we typically remain protected against the disease for years, for decades, or even a lifetime. This is what makes vaccines so effective. Rather than treating a disease after it occurs, vaccines prevent us from getting the disease in the first place. So important to remember that usually, and of course this varies by vaccine, we have protection for a long period of time, and it can be a few months, it can be a few years. It depends on the nature of the vaccine and the type of disease we are dealing with. Occasionally needs what are called boosters, so repeat vaccinations just to strengthen and the memory of the immune response. And for certain vaccines you need several doses, whereas for most vaccines you really just need a few doses for them to work. So as I mentioned earlier, there are different types of vaccines. So there's this Thermolive attenuated vaccines, and attenuated just means that it's a weakened form of the organism itself. So it's, in the case of the measles vaccine, they've taken the measles virus and they've gone through a laboratory process with that measles virus and made it weak to make sure it doesn't cause disease. So they've changed the virus and it's referred to as a live attenuated vaccine. Then there are vaccines that are made up of the whole cell of the organism, be it the virus or bacterium. And the example here is whooping cough. So this is not a live vaccine at all, but it's the whole cell that's been killed, but it of course still retains the antigens that induces an immune response. Then there's our viral vectored vaccines, and we've heard a lot about that amongst the COVID vaccines. So these are viruses that have been adapted, and we use the term vector because it carries a particular part of the germ that we're interested in. So there's a whole range of viral vectored vaccines. So there's one that's been produced for Ebola, for example. I've given the J&J COVID vaccine as an example here because the virus that's used in the Johnson & Johnson vaccine is the adenovirus, and it has in it the code for the spike protein of the coronavirus. And that's how it works. But the virus in this case acts as the carrier of the code for the antigen that's going to induce the immune response. Then there are protein-based vaccines where the actual spike protein, for example, like Novavax, is used as part of the vaccine to directly induce an immune response. There are DNA-based vaccines, so the hepatitis B vaccine that's been around for several decades now is DNA-based. And then what we've heard about more recently, and this is relatively new technology for the Pfizer and Moderna COVID vaccines, the mRNA vaccines, and mRNA simply stands for messenger ribonucleic acid. And it's really a mechanism by which our cells produce proteins. And when messenger RNA gets into our cells respond by producing a protein. And these mRNA vaccines of Pfizer and Moderna, when they get into the body, our own cells actually produce the spike protein of the coronavirus. Now, of course, the spike protein on itself, on its own, cannot cause any harm, but it does induce an immune response. And that's how it works as a vaccine. Right. So how are vaccines developed? So as with all medicines, every vaccine must go through an extensive and rigorous testing process to ensure it is safe before it can be introduced to any country. And, you know, there's been decades and decades of science around this process of developing new products, new medicines, and the methodologies have improved, the ways of actually doing such clinical trials and testing has been honed and refined over many, many decades, which is why we are at the point where, you know, when we test new products like vaccines, there's a very thorough process, but also a regulatory process. So I'll talk a bit about the regulatory side a bit later, but just to understand, there's a strong scientific process behind product development like vaccines, and it's aimed to ensure that whatever medicines are used by the public are both safe and that they work. Often a lot of this happens behind the scenes. Most of us are not even aware of the processes that new medicines go through before they make it to market. Often an experimental vaccine is first tested in animals to evaluate its safety. So this is another safety aspect of vaccine or any product development. It is then tested. So I've just put a note at the bottom there that before we get to the experimental vaccine, laboratories where they take samples of the organism itself and they develop experimental vaccines based on what they know about what could induce an immune response. And these experimental vaccines then are first tested in animals before they are tested in humans. And when a product or vaccine is tested in humans, it's tested in the form of clinical trials. But there are three types of clinical trials, or three main phases. In phase one, the vaccine is usually given to a small number of adult volunteers to assess its safety, confirm it generates an immune response, and determine the right dosage. So usually there's about 20 to 30 adults who are usually healthy, and they usually go through several tests to ensure that they are healthy. They go through consent to make sure that they understand that they, this new product or the vaccine is being tested on them at phase 1 study. You go to phase 2, where the vaccine is usually given to hundreds of volunteers. They're closely monitored for any side effects, again for safety and to assess its ability to generate an immune response. Sometimes in phase 2, data is also collected on possible disease outcomes. So you sometimes have reference to a phase 2A or a phase 2B. So the phase 2A is purely based on safety, but in the target group that you're interested in, if it's children or if it's HIV positive people or if it's adults or the elderly. In phase 2 is when you test it in your target group and you test it for safety. But when you get to phase 2B, you actually start looking at whether this vaccine works, but you usually have what's called a proof of concept trial. So the numbers are a bit bigger so that you can evaluate efficacy to see whether it works or not, but usually not enough to register the product for public use. So just to understand that there is a difference between what's called phase 2A and phase 2B. So usually by the time you get to phase 2, you have a control group. So some volunteers get the vaccine and others do not. And that allows comparisons to be made and for conclusions to be drawn about the vaccine. In phase 3, the vaccine is usually given to thousands of volunteers. Some of whom receive the investigational vaccine, some do not. We often talk about the placebo group or the group that gets something that doesn't have any effect. But please keep in mind that sometimes, such as in the Oxford AstraZeneca vaccine, you give the control group a different vaccine or some other product which may have some benefit. And sometimes it's useful to do that to ensure that your two groups are comparable in terms of the experience of being vaccinated. We still look at safety. So safety information continues to be collected in phase 1, phase 2, and phase 3. But by the time you get to phase 3, we talk about phase 3 as a registration trial, because here the attempt is to do as much testing in a large sample of people so that once this trial is done, you are able to register it for use by the public. Sorry, just having something to drink. Yeah. So I think before I go off this, people often talk about the phase 4. And just to mention in passing that phase 4 usually refers to vaccine testing after a vaccine and is already being used in public. Sometimes, let's say, pregnant women or HIV positive people, there might be particular groups that you might be more interested in, and that would be what's called a phase 4 type study. There's often this distinction between vaccine efficacy and vaccine effectiveness. And not to get too technical about this, vaccine efficacy is when something is done under very controlled conditions, whereas when you test something in the real world or under real life conditions, it's often referred to as an effectiveness study. But that's mainly a technical definition. You may or may not come across it, but often these two terms, efficacy and effectiveness, are used interchangeably. So, how do we actually do a vaccine efficacy study? So as I've mentioned, it's a phase 3 trial. So just to explain these terms, double-blind, randomized, controlled. So double-blind refers to the fact that the people giving the vaccine, the investigators, and the people receiving the vaccine don't know whether they're getting the vaccine, whether they're getting a placebo or a control product. And that's what double-blind means. That's that neither the subjects or the participants in the trial nor the investigators are biased. So when they evaluate whether someone gets COVID or not, for example, in a COVID trial or any trial, when you evaluate whether someone has the disease or not, you're not influenced by the fact of whether you know whether someone has had the vaccine or not. So to reduce unconscious. This is an important principle of science that you find in clinical trials, is that whoever gets the vaccine or gets the placebo product happens in an random way. So you cannot tell in advance. And there are various sort of computerized techniques of doing it, of randomizing. So this will not be alternate. So the first person gets the vaccine, the next person gets the placebo, and so on. That wouldn't be randomized. Randomized is when you just don't know in advance each next person is going to get either the vaccine or a placebo product. And this again is an important scientific control to ensure that we cannot predict as the investigators who has had the vaccine or not, and also to balance the groups, so to balance those who get the vaccine and those who don't get the vaccine. So what do I mean by balance? It means that the profile of the people who get the vaccine and those who don't are the same. They have the same age structure, they have the same gender structure, they have the same geographic area structure, or any other important characteristics are equally distributed between the different arms of the study. And that's very important in the randomization, so that when your results come out, you can be sure that the groups being compared are fairly compared, that they are the same, that they're not different in any way. And controlled simply means that there's a control group. So in the graphic at the bottom, you'll see that you have a sample population. They are randomized either to treatment A or to treatment B. Treatment B might be your placebo or control group, and you then follow them up for outcomes. So treatment B in this case would be the control part, or the placebo control group. So conditions are idealized normally when you do a clinical trial. There's often strict inclusion and exclusion criteria. So for example, you'll very clearly say pregnant women are not allowed, or HIV positive persons are either allowed or not allowed in the trial. People must sign consent before they can get into the trial, and so on. So there are often specified criteria as to who can be in a clinical trial or not. The sample size is determined based on what's the expected incidence of disease in the unvaccinated group is and the estimated incidence in the vaccinated. So to simplify this a little bit more, you are looking at the rate of disease in the community, if you like, or in which your unvaccinated group will represent, and you kind of guess what you think you're going to achieve by reducing the disease risk in the vaccinated group. So your sample size then is determined. So this is one of the important factors. I think another factor to mention is that your statistical power, which is important in a phase 3 trial, is whether whatever result you get, you can be confident within a narrow interval that this is a genuinely valid result. So sample size is extremely important in clinical trials, or the sample size here meaning the number of people participating in the clinical trial. And then, of course, a clinical trial is run for a specified period of time. I'm going to go through an explanation for how one calculates vaccine efficacy. If you're not comfortable with mathematics, you may not be comfortable with this part, but I've tried to keep it as simple as possible. And it's important just to help us understand when we see these figures, 95% efficacy, 60% efficacy, 70% efficacy, what does it actually mean? So in this example, you've got in the group that got placebo, 100 out of 1,000 developed disease. In those who got the vaccine, 10 out of 1,000 developed disease. So you can see the rate of disease in those who got the vaccine is less. So you can see that there was a benefit here. So just on the face of it, you can see there was a benefit to those who got the vaccine. And again, of course, you're assuming that this trial was conducted with all the necessary correct procedures in place. So how do you calculate then the rate of the vaccine efficacy? So we look at the incidence of disease in the unvaccinated. You subtract the incidence of disease in the vaccinated. You divide that by the incidence in the unvaccinated and you multiply by 100. So this is all technical to you, so let me just explain it. As we saw on the previous page, 100 out of 1,000 got the disease in the unvaccinated group, minus 10 out of 1,000 got disease in the vaccinated group, divided by 100 out of 1,000. And if one takes this further then, so that you've got a 10% risk of developing disease in the unvaccinated group, 1% risk in the vaccinated group, divided by that 10% in the unvaccinated group. So 10 minus 1 gives you 9 over 10, and you create a percentage out of that, and essentially you get this figure of 90%. So this vaccine, in this theoretical example, was 90% efficacious. But what does this mean in simple language? All it means is that you had a risk of 10% if you were not vaccinated. You had a risk of 1% if you were vaccinated. So the reduction in your risk was 90%. Your chances of developing disease if you were vaccinated was reduced by 90%. And that's fundamentally, in simple terms, what vaccine efficacy is. So what is the public value of vaccines? So vaccines have been one of the most effective health interventions after the provision of safe water. It has saved millions of lives. There's suddenly a lot of interest in vaccines, almost as if it's something new, but it's part of the normal fabric of society. It's been around for decades. We give them to our children. We use them when we are injured, for example, the tetanus vaccine. And for those of us lucky to travel, there's a yellow fever vaccine. And interestingly enough, with the yellow fever vaccine, it's the one vaccine that's legally required. If you travel to a particular country that's a yellow fever country, you cannot, for example, from South Africa, you cannot return to South Africa without having been vaccinated against yellow fever. It's compulsory. And then, of course, smallpox, which some of you may or may not remember, has been eradicated from the world. It was eradicated in the late 1970s due to smallpox vaccination. So this is an amazing achievement of vaccination, that we've actually eradicated. We've got rid of a particular disease from the whole world due to a vaccination program. And then, of course, polio, which is a disease that causes paralysis, only exists in a very few countries, mainly in Asia at the moment. Again, due to a global vaccination program. It's been absolutely amazing what polio vaccination has achieved. And we can repeat this picture for many, many other diseases which have become fewer and fewer over time. You know, we see very few measles cases now in our health system, and that's due to the large-scale measles vaccination programs that have been in place. We saw a huge measles outbreak in New York a year or two ago, and that's because, at least in part, due to measles vaccination going down in countries like the US, you know, a stronger anti-vaccination sentiment developing and fewer people being vaccinated and therefore more people being susceptible. But measles as a disease, although it can be coped with by many people without any major issue, can be quite a serious disease, particularly for poorer people or for malnourished children who can die of measles. I don't expect you to read all of this, but this is the childhood immunization schedule in South Africa, and there are 10 different vaccines on this schedule, and this is what children normally get on a daily basis. And these are all provided free in the public sector in South Africa, and in many African countries there are similar schedules. Again, just to show the impact of polio vaccine on polio incidence. So the red line shows over the last 40 years dropping incidence of polio as vaccination coverage increased. So coming to COVID-19 vaccines. So we've set these goals. We want to prevent severe illness and death. We want to reduce transmission, and we want to protect our health system. So the first and the third goal we can start doing fairly soon, because as soon as someone's vaccinated, and I'll talk more about that in due course, there's a good chance we'll prevent them from developing severe disease and from dying from COVID. And because we are targeting health workers first, and the World Health Organization is very much supporting this goal of let's vaccinate our health workers first, we protect our health systems so that we can care for the people who are developing COVID. The issue of reducing transmission is more tricky. Ultimately, with many diseases, we want to achieve what's referred to as herd immunity. With COVID, we are not sure about when we'll be able to achieve this. There's only some data on the reduction of transmission due to vaccines. There's the issue of variants, which you will discuss in another talk. So there are a number of factors that make it difficult to predict how successful we'll be in reducing transmission. So we'll have to watch that space. Important to mention, as of a couple of days ago, almost 300 million vaccine shots for COVID-19 had been given around the world. And there's this particular website that I've used to get that information. Most of it in particular countries, mainly in wealthy countries. And we all know about the so-called vaccine nationalism. When I got this data, this is from about a week or two ago, 130 countries had not yet started administering a single dose. South Africa is in a fortunate position having to have just started the vaccination program. But there's clearly been a maldistribution of vaccines around the world. This might be a bit small for many of you, but these are the 8 vaccines that have shown results. So on the left, you'll see the Pfizer vaccine, Moderna, Oxford AstraZeneca, also known as Covishield, a vaccine produced by Sinopharm, which is a Chinese company. Another Chinese company, Sinovac, has produces a vaccine called CoronaVac. Sputnik 5, we know, is a Russian vaccine. Novavax, also produced by an American company. And at the bottom, the Johnson & Johnson vaccine that we know about. If we look at the efficacy column, so this is vaccine efficacy. So for example, you'll see there Pfizer had a 95% efficacy in their trial, Moderna 94%. Oxford AstraZeneca had variable results between 62 and 90%, tested in Brazil and the UK. And unfortunately in South Africa, it had a 22% efficacy, so not really effective in our context. Sinopharm 86% and so on. The Sputnik 5 91.4%, and at the bottom there you see Johnson & Johnson 66%, but varying by country between 57% and 72%. Interestingly enough, the second column looks at severe disease, and I'm not going to read out those numbers, but what you can see is a very clear pattern that with virtually all these vaccines, compared to their overall efficacy, generally efficacy against severe disease is much better, and in many instances 100% effective. And this is really exciting information especially for that first goal of vaccination, which is to reduce severe disease and mortality or death due to COVID. And I think this is one of the best parts of the vaccine development program, is the ability of most of these vaccines to prevent severe disease and death due to COVID. Efficacy in high risk groups, again, some vaccines have managed to show this. So in diabetics or hypertensive, people with comorbidities, in older age groups, I see the Johnson & Johnson block is empty there, but they have in fact tested their vaccine also in older age groups and in people with comorbidities. The dose regimen, you can see for most of them it's 2 doses, but the Johnson & Johnson is the exception with 1 dose. Storage requirements, again, you'll see with Pfizer, they have difficult storage requirements. You need a -70 degrees freezer. With Moderna, -20 degrees. Many of the other vaccines, though, you can store at 2 to 8 degrees in a normal vaccine fridge, which make them more easily available to countries in Africa. The Johnson & Johnson vaccine is normally stored at -20 degrees, but can be kept in a normal fridge between 2 and 8 degrees. In the last column, I've put whether these results have been published in scientific journals. And again, for me, it's important to have these results being checked through a scientific review process. And it helps if it's been published in a journal. For Johnson & Johnson, they've made an submission to the FDA. And of course, they'd have to publish detailed data. And based on the FDA submission, they were given emergency approval by the FDA in the US. So some of these results have been published in scientific journals, but others not. Right, so talking about approval processes. So once a vaccine has been shown to be efficacious, they often want to now register it for public use. And there are different mechanisms for this. So many countries have what are called medicine regulatory bodies that approve both the clinical trials of these new products, and there are ethics committees that then also have to approve the research. So before we actually even get to approval for use by the public, the research itself also has to be approved independently by these regulatory bodies and by ethics research committees. So there's an independent checking process. And obviously, there's a big responsibility on these regulatory bodies and ethics committees to ensure that the trials are properly designed and that they are safe. And that there are also proper safety reporting mechanisms in place. And once that's done, if a trial produces efficacy results and the organization that's developed the vaccine wants to register for public use, there are 2 mechanisms. There's a normal approval mechanism that normally takes several months before an approval happens. And then there's what we've commonly seen now with COVID, an emergency approval. So that's a rapid approval based on a rapid review process. So usually there's an extensive review process which provides then approval for general use, and there's an emergency approval. So most of the approvals that we've seen around the world have been emergency approvals. And that's for the very reason that we've got an ongoing pandemic and there's an urgent need for the use of these vaccines. Again, I've just emphasized at the bottom there, these approvals must be independent of the researchers and pharmaceutical companies. So that's an important part of the process. And again, this is where a lot of questions are asked about these vaccines. Are they, are there conspiracy theories about them, or have they just been, developed for profits by various companies and so on? But there are meant to be these independent full approval processes to make sure that these vaccines are genuine, that they follow the correct procedures, and that they can be used safely by the public, and that they work for the public. So just some examples of regulatory bodies. In South Africa, we've got the South African Health Products Regulatory Authority, or SAPRA. In the US, it's the Federal Drug Administration, or FDA. In the UK, it's the Federal Health Regulatory of the Medicines Health Regulatory Authority, or the MHRA, and in Europe, it's the European Medicines Agency. And then, of course, the World Health Organization also acts as a regulatory authority and provides approvals for and guidelines for many of these vaccine products. So I've come to the end of my presentation. So just to give some concluding comments. So vaccines have played an enormously important role in the control of infectious diseases and continues to do so. So they've been extremely valuable to date. There is a rigorous scientific and ethical process underlying the design, researching, and implementation of new vaccines. 8 vaccines have shown efficacy against COVID-19, and some of them have been approved for use in multiple countries. So I should just qualify that approval process. Not all of them have been approved as yet. However, I think one point to mention is that vaccine distribution inequity remains an important stumbling block to global control of COVID-19. Thank you.
Thanks, Hassan, for your presentation. We will now have opportunity for questions, and I've noted that there's been quite a few questions already being put in the chat. I'll hand over to Anal to just pose the first 3 questions to Hassan. Anal, over to you.
Hello, Professor Hassan, and thanks everyone for the questions. Firstly, Kevin Brandt from Eyewitness News asks a question in the context of people in South Africa who've been reinfected with COVID-19 without having been vaccinated. He wants to know, what is the difference between the human body mounting an immune response after a viral infection and the immune response generated by vaccination? Is the response generated by a vaccine better? And if there is a difference, how are the 2 versions of immune responses different? Okay. That's the first question. Secondly, Jemima Achivers, she's a freelance journalist from Ghana, and she wants to know how long would the vaccine remain effective from when the body begins building immunity after it's injected with a vaccine. Okay. And then thirdly, Victor Mulangise is asking, do you see Africa being able to reach herd immunity this year? And if not, what are the drawbacks and how can they be overcome? So Professor, just to make it easier, if it will make it easier to you, I will also just pose these questions now in the chat again if you need to relook at them.
Yeah, thanks so much for those very useful questions. And the first question by Kevin Brandt. Yeah, so with COVID, with getting the disease, we do know that there is a risk of reinfection. So there is a chance with natural infection that your immunity wanes, and that therefore there is this risk of developing a second infection. At the moment, there are not that many proven cases of reinfection, but certainly there are a few, and they've been proven, so that remains a risk with natural immunity. And again, we are learning and we don't have all the answers. So for many diseases, natural infection can sometimes be enough to provide protection for a long time or even lifelong. But for COVID, it's uncertain and unknown. So I think it's more a question of being uncertain about the length and the degree of natural immunity, which is why vaccination is still recommended worldwide, even if you've been previously vaccinated. So the vaccine usually focuses on one protein or one element, which is designed along that route, and that's what induces an immune response. And of course, it's a safer way of inducing an immune response, and that's the main difference. So, I mean, the second question is related to this, in that we don't know how long vaccine immunity will last. And there are two factors there. The one is that in certain instances, vaccine immunity wanes over time and one needs to give a booster. So it's an advantage giving someone a booster, excuse me. It's safer to do that, to achieve protection rather than letting someone remain at risk of developing a second infection, which may be potentially quite severe. So it's more from that aspect that one encourages people to get vaccinated. So if boosters are needed, we can continue to give them in a safe way without causing any harm, whereas we wouldn't obviously want people to develop repeat infection. However, the one confounding factor, if you like, is this whole issue of variants and whether the virus will continue to adapt and reach a point where these vaccines do not work. And then that puts one at risk of developing another COVID infection where the vaccine that you've been given is not effective against that particular variant. So the science is evolving, it's developing, and there are a number of issues that are still not certain. But I think just in summary to say, there's still uncertainty about the length of time that natural immunity gives you protection. So that's a concern. But we are also uncertain about the length of time that vaccines give you protection. But it's a safer way of inducing immunity compared to getting the disease naturally. So that's really the advantage. But the confounding factor, I would say, is the whole question of variants and how that might impact on ongoing protection against COVID-19. So I think that's a space that we need to watch on an ongoing basis in the future. And we may, in fact, have to adapt our vaccines as the pandemic evolves if these variants become significant and impair immunity. We know that the so-called South African variant has influenced vaccine efficacy. So from 3 different vaccine trials done in South Africa, Novavax, Johnson & Johnson, and AstraZeneca, the vaccine effect was less or no effect in the case of the Oxford AstraZeneca vaccine, but less in the case of Novavax and in the case of Johnson & Johnson compared to vaccine efficacy in other countries. So we know these variants are important, and it's something that we need to watch further. The question of herd immunity is a challenging one, and my simple answer to you is no. Okay, it's highly unlikely that we were able to achieve herd immunity, but of course, I am estimating this based. I'm giving an opinion based on current conditions, and that is the current supply of vaccines. The current supply of vaccines is very slow and is coming in drips and drabs. And as I understand, for example, like the COVAX facility, which you'll hear about more in a later talk, the amount of vaccines that we will be getting through facilities like COVAX or other means is nowhere near enough to achieve what's referred to as herd immunity. And I think I want to emphasize that herd immunity is about getting between 60 and 75% of the population immune, okay? Not vaccinated, immune. And because these vaccines are not 100% effective, we actually need to vaccinate a much higher proportion of the population to achieve that level of immunity. So the percentage of immunity required for herd immunity seems to vary quite a lot around the world, but it's in that range of 60 to 75%. But to achieve that will be a massive task. And even in those first world countries, they may also struggle to achieve herd immunity, depending on the vaccines that they use and depending on the variants that might occur in the future. So I think this is an issue for the world and not just for Africa. Thank you.
Thank you, Professor. So I'll read the next 3 questions. So Natalie Rietgaard from UNAIDS, she's asking, why is there different efficacies in different countries with the same vaccine? Okay. Then Apumulele Mdlalane from South Africa, he's asking, what happens when one has taken a vaccine and then there's a new variant that's identified? Will a person have to take yet another vaccine in order for them not to be infected with a new variant? And then Moses Ziyambi, he's asking, does WHO have the final say on all vaccines introduced onto the global market? And if not, is it safe then to trust vaccines developed in those not so open countries such as China? I'm going to post the questions again in the chat.
Thank you so much for those very interesting questions. So the first question, again, I think I've partly answered that, different efficacies in different countries. So the one clear answer to that question is the existence of different variants. So the so-called South African variant in South Africa has vaccine efficacy in other countries, and there may be other variants. Let me just say one thing about variants. There are hundreds of variants actually of COVID, and most of them are completely harmless. And that, will help answer some of these other questions that have come along. So that's one of the main reasons. There may be other reasons. It may be due to particular population factors or conditions in different countries. The degree of infection from other diseases. For example, some of these vaccines use adenovirus. And if that adenovirus is present in your population, it may impact on your vaccine efficacy. But generally, the adenoviruses used in vaccines are not that highly prevalent. So there may be other factors that may be at play that influence vaccine efficacy and the variation between different countries. But for the moment, what's been the most obvious factor or most clearly demonstrated factor is the existence of these variants impacting on different efficacies in different countries. So will you need another vaccine if a new variant develops? So as I've said, there's several hundred variants. So just simply the existence of a variant doesn't mean you need to take a new vaccine. That variant will have to be evaluated against the existing vaccine to see whether it works or not. And there are ways of doing it. So the best way of doing it is in a clinical trial to see if the vaccines remain efficacious. But for example, Pfizer's tested their vaccine against the South African variant in the laboratory using more artificial means of testing whether it works against the new variant or not. So there are other techniques that can be used. So that would be the first step to see whether vaccine efficacy is impacted on by new variants. And only if that is shown to be the case would one have to consider additional or different vaccines. So, for example, with the Oxford AstraZeneca vaccine, when we got the results from the South African arm of the study showing very little benefit here, the developers of the vaccine said that they will work on a new version of their vaccine, and it will take up to 6 to 7 months to develop such a new version that will be effective against the new variant. So it's possible for existing vaccines to be adapted, and that's an area of development. I think the role of variants is being studied very carefully, and it's something that we do need to be concerned about going forward, about whether variants will continue to affect us, and needing us to actually change or adapt our vaccines, similar to the way we have to adapt the flu vaccine annually, to keep up with the change in the strains. Are we going that direction? It's not certain at this point in time, but certainly our latest experience with the Oxford AstraZeneca vaccine is of concern, and that's something we need to monitor more carefully going forward. Does WHO have the final say in terms of the global market? Definitely not. I think any country can make an arrangement with a vaccine manufacturer for the delivery of vaccines and for approval of vaccines. I think what's more important, though, is to ensure that you have a strong regulatory authority, and that it's possible in many countries that they don't have strong regulatory authorities, and this is where WHO plays an important role in providing that strong regulatory control focus. South Africa also depends on the regulatory authorities that in more well-resourced countries like the FDA, like the MHRA, like the EPA in Europe, those regulatory authorities are better resourced, and they are able to perhaps conduct the necessary thorough scientific reviews, and we can then use those reviews to help us, not necessarily automatically, but it can assist us in making decisions about emergency approval and so on. And our South African Regulatory Authority definitely does that. We use authorizations obtained through other regulatory authorities to help us decide locally, and not necessarily wait for the WHO to make a decision. Nevertheless, WHO can play an important role where countries are concerned that they don't have the necessary regulatory resources in order to properly evaluate vaccines. And then I would encourage those countries to wait for WHO to provide guidance on these matters. Thank you.
Thank you, Professor. Next 3 questions. First one from Victor Mulangisi. What are the global advantages if there are different vaccines from different countries? Won't this compromise the uniformity of receiving equal effective treatment? The next one has a similar theme. So that comes from Natalie Rietgert from the UNAIDS. She's asking, with the early vaccines, did we see the same access issues and inequalities as we have seen with HIV treatment and now with the COVID vaccines? And then the third question in this batch comes from Busani Bafana from Interpress Service. He's asking, how watertight is the emergency approval of COVID-19 vaccines, given that it has taken a long time for the approvals of other vaccines such as TB? Are emergency approvals safe? I'm going to post these again now in the chat.
Thanks so much, Anal. And again, thank you for these, very interesting questions. So, I mean, there are definite global advantages to having different vaccines. That's, of course, an issue of supply. The big issue globally is supply of vaccines. So having multiple manufacturers producing vaccines, having multiple, I mean, certainly one or two vaccine manufacturers cannot supply vaccines to the world. So if you have multiple vaccines available, The World Health Organization has set a fairly low level of about 50% efficacy for a vaccine to be acceptable. And, all the vaccines that I've listed have met that standard. And they are then available to be used. I think the important part of the vaccine experience is that its efficacy against severe disease. So there's certainly that advantage where, in order to reach or achieve global needs, one can draw on then multiple vaccines to meet the global need. And then some of the vaccine manufacturers, or many of them, are sharing the technical know-how with other vaccine manufacturers. So I think it's Moderna or Pfizer in the US. I think it's probably Moderna that's sharing the manufacturing capacity with Merck. So they're both going to be able to manufacture the same vaccine. Subject to correction on that, but there've been partnerships between different pharmaceutical companies to produce vaccines, and that's obviously an amazing advantage. However, as Natalie raises, issues of access to these needed medications, the access to patents, that's still an ongoing issue of debate internationally, and providing that know-how broadly around the world. There's still some, stumbling blocks to achieve that, and that somehow impacts on the availability of vaccines. So a lack of willingness to just simply share patents for the global good. So the struggle around antiretroviral medications, there's a history of that where activists had to fight to achieve better access to drugs. There should be a similar fight for COVID vaccines, in my view, because this is a global pandemic. And I must say I am at one with the WHO Director General when he says no country is safe until all countries are safe. We really should be doing this collaboratively. And, the vaccine nationalism that we're seeing is really not in the interest actually of any country in terms of achieving control. Is the emergency approval process watertight? So, okay, it's difficult for me to give you any certainty on that. Obviously, it's a more rapid process, so it's less perfect, but the way it's implemented is with more tighter controls. So from what I've seen of the emergency use process in South Africa, you would need a consent process. There are particular reporting requirements that go with it. So it's not as tight as your normal approval processes, but there are more stringent controls with the implementation of emergency use from what I've seen. So there's attempts at bringing in sort of some controlling balances into the process of emergency approval. It's not simply go forth and use the vaccine in whatever way you want. So most of the vaccines that we are using on a daily basis don't require a consent process. They have very limited reporting requirements, whereas those requirements are a lot tighter with an emergency use approval. So that's all I can say, is that there are more stringent control measures associated with emergency use that may not necessarily imply that the review process is necessarily tighter. But from what I've seen, many countries have adopted this. They've weighed the risks and the benefits, and clearly they see that the benefits outweigh the risks, because we are in the middle of this massive global pandemic, which is wreaking havoc, causing morbidity and mortality, damaging our health systems and so on. So that is more the discussion, rather than trying to attempt to answer the question of whether this is watertight or not. And in fact, any regulatory process will not be completely watertight. And even after approval of any new product, there's always ongoing monitoring, post-registration monitoring, adverse event monitoring, etc., that continues, because no process is, of course, completely watertight. But that's as far as I can answer that particular question. Thanks.
Thanks, Prof. I think we have time for 3 more questions. First one comes from Sunay Payne from the Daily Maverick. She's asking, from a health perspective, how would you describe the media's reporting on COVID-19 and related issues? And where do you think we should better or to focus better on? Then secondly, from Aisha Abdul Karim from Bhekisisa, she's asking, why did the AstraZeneca researchers opt to use the meningococcal vaccine as a comparator arm instead of a placebo in 2 of the phase 3 trials? What are the benefits of using such a comparator in this scenario when it wouldn't provide any additional protection to the participants? And then lastly, Kate from MSF in South Africa is asking, despite prohibitive barriers like cost and cold storage, is an mRNA vaccine like that offered by Pfizer the most ideal option, as it can be tweaked for variants? Also, is it possible that the first shot has one kind of makeup based on trial data, etc., and the second one is adapted to the variant? I'll post the questions again.
So thanks so much. Thanks for those questions, and thanks for asking very important and very useful and very challenging questions, I must say. In terms of media reporting, again, for me, it's, I actually think that media reporting has been quite good. And obviously, this kind of course is useful for journalists to be on, to make sure that they are, properly informed about, the COVID-19 pandemic, about, vaccines, to understand at least some of the technical issues, scientific issues behind these vaccines. But generally, the reporting has been impressive and has been critical at times, but it's been usefully critical to raise many, many key issues. I mean, I think on occasion there is, of course, misinformation that goes by the media, but most of the misinformation has been spread rather through informal means rather than through the formal media, and that's generally been my experience. My experience of the general media or the formal media that I've had dealings with has been generally quite good and well informed. So I don't have any particular concerns, and it's quite I think obviously helpful that you as journalists are engaging in informing yourselves of some of the issues and challenges that exist in the vaccine arena through this mini course and through other courses that you've engaged in. The Oxford AstraZeneca vaccine using a control vaccine. Yeah, so I mean that sometimes one can argue, or sometimes ethics committees argue, that it's useful to give, rather than give the control nothing, to give them something. The meningococcal vaccine that was given has no benefit. It obviously has a benefit in terms of preventing meningococcal or meningitis as a disease, or the particular organism that was used in that particular vaccine. There may be a technical advantage that because that person's getting a vaccine, the responses that you get after vaccination are similar to what you'd get after the actual COVID vaccine. So you can't really tell the difference, and because of that, you can't somehow I suppose, inadvertently pick up who has actually received the vaccine or not. So there may be technical reasons why they chose to do it, but why, for example, they chose to do it in the UK and Brazil, but not in South Africa, that is not a question that I can answer, and perhaps the researchers can perhaps try and answer why there was some difference. Possibly the regulatory authorities or the ethical authorities might have asked for those variations. But I think please remember that the Oxford AstraZeneca reports that have been published are combinations of trials. So the trials, there are 2 trials in the UK, 1 in Brazil, 1 in South Africa. They're 4 different trials, and in the reporting of these trials, they've combined the results. So it's a little bit different from some of the other trial results like Pfizer and Moderna, where the whole trial was one trial but conducted in different countries. So it's a slightly different approach. But I don't think it was necessarily, and so the meningococcal vaccine is that they use is obviously an established vaccine with an established safety record. So the benefits obviously would have been seen to be outweighing any risks potentially of giving that as a control measure. So as I said, there might be technical reasons for that. And then in terms of the mRNA vaccines, does it have an advantage in terms of being adapted for variants? Yeah, so I mean, as you correctly indicated, has quite stringent cold storage requirements. Hopefully with further research, they can address some of those storage requirements. Can it potentially be tweaked more easily? Yes, potentially so. For variants, just please note that Pfizer, when they assessed the efficacy of their vaccine against the so-called South African variant, they didn't tweak their vaccine. They kept it the same, and they just tested in the laboratory to see whether it actually worked against this new variant. But because it's a molecule, it has the potential to be adapted perhaps more easily. So one have to wait and see. And one has to look at the number of factors. The storage requirements, one, this vaccine is a lot more expensive. These Pfizer and Moderna are a lot more expensive than some of the other vaccines. And there's also, it's a relatively new technology. There's not as much experience with it compared to other vaccines. So there may be pros and cons to their approach, but if it evolves and develops and becomes established, it may be a better way in the future of developing vaccines. So that's the only answer I can give, which is a fairly broad answer to your particular question. Thanks.
Thank you, Prof. We've got 4 questions left in total, but we won't have time to get to them all. Maybe I can just ask one more quick question from Johnny Isaac. Is there a need that we take the flu vaccine? Just for the sake of time, I'm just going to ask one question, short question
yes,
at a time.
Absolutely, yes, we must. Yeah. So, but important, as I've been asked many times, if you take the flu vaccine, you are not protected against COVID. So there is a little bit of confusion around that. But for those people who are at risk of severe flu, should still take the flu vaccine. The COVID vaccine will not protect you against the flu. The flu vaccine will not protect you against COVID. They're 2 completely different viruses, 2 completely different diseases. So both vaccines must still be taken, as needed. So the COVID vaccine, ultimately everyone is eligible for that, and the flu vaccine is recommended for particular high-risk groups. Thanks.
Thank you. One last interesting question. From Kevin Brandt. If a vaccine has expired, is it literally on that day and not one day later? And what is the concern with this expired vaccine? Is it efficacy that goes down, or does it become toxic for human use?
Yeah. It's probably highly unlikely that it's, one day it's working and the next day it's not. So the way these vaccines are tested is, they test it over a long period of time, and they test viability on a daily basis to ensure that it remains viable. So at that point where it expires is when the vaccine starts to become, less viable, less efficacious, or in terms of content. So there's probably a slow change in vaccine efficacy, but it starts dropping to a level which is not acceptable from a vaccine quality point of view. So there's never a sudden change. I think it's unlikely to be regarded as poisonous. It's more likely that it slowly becomes no longer efficacious. And it's just due to ongoing sort of daily monitoring under normal storage conditions that these products are being tested. And it's really about at what point do they start decreasing in its content, where one is concerned that they're no longer infectious, they're no longer effective, and they would presumably set that bar at quite a high level. So any indication of deterioration in the quality of vaccine, would be of concern. But I doubt that it would be regarded as poisonous, but rather that it's no longer efficacious.
Thank you. And perhaps for a 1-minute answer, can you switch from one vaccine to another after the first dose?
Yeah, so there's ongoing research about that. At the moment, what's been advised is that we don't do that, but that is being investigated, that you can change from one dose. And sorry, that was part of a prior question where someone asked, if you took one dose of the vaccine and you had a new variant, could you take a changed version of that vaccine later? So presumably yes, but mixing vaccines at this moment is not advised because we don't know enough, not because it's harmful. It's just that we don't know enough about what the impact may be. But it's not that there's a specific reason that it might be harmful. It's more a question of not knowing enough at this stage, and it's for the moment not advised. Thanks.
Thank you, Prof. Professor Young, over to you for wrap up and closure, please.
Thank you. Thank you for facilitating the question and answer session. Big thank you to Hassan, who has joined to set the scene for our first out of 4 sessions for this course. Hassan focused on vaccine development, on covering how vaccines work, how vaccines can be tested to see whether it's safe and whether it works. Also the whole process of getting it registered for use in country. So huge thank you again, Hassan. You've really set an excellent scene for us for our follow-up questions. Our next session we will be having on Thursday, this coming Thursday at the same time at 7, and we'll be focusing on vaccine implementation. And next week, Monday, we're focusing on coming back to the big issue of vaccines and variants. And next week, Thursday, we'll be covering COVAX. So we look forward to all of you joining us again this coming Thursday, where we will have Richard Mehigo from the WHO Regional Office, who will take us through vaccine implementation. But for now, I want to wish all of you a very good evening. Enjoy the rest of the week, and we will see you on Thursday evening. Thank you all. Goodbye.